We performed a proteomic screen to identify novel interactors of RPA, an essential single-stranded DNA binding complex with critical roles in DNA replication and repair and in DNA damage signaling. We used biatylated single-stranded DNA and coded it with recombinant RPA complex. RPA-coded single-stranded DNA mimics the structure that is recognized by the ATR ATRIP kinase and other proteins to elicit the DNA damage response. We then exposed RPA-coded single-stranded DNA to nuclear extracts, and the interacting proteins were identified by mass spectrometry. To identify novel proteins important for DNA damage signaling, in addition to wild-type RPA, we also used a T11 RPA mutant. T11 is a separation of function mutant fully proficient for DNA replication, but defective for DNA damage signaling. This defect is caused by a mutation in RPA70, which abrogates its interaction with ATR ATRIP and other critical regulators of the DNA damage response. In contrast, the eligase Marechal1, which binds to RPA32, interacts equally well with wild-type and T11. In panel C, we separated the putative RPA interactors by SDS page. This shows that wild-type RPA interacts with more proteins than the T11 mutant. We expected that the subset of proteins that interact with the wild-type RPA complex but not with the T11 mutant would be highly enriched for regulators of the DNA damage response. Panels D and E show that our screen worked, as the most abundant category of proteins identified in our screen were those implicated in DNA replication, recombination, and repair. Amongst these proteins were many non-interactors of RPA. As expected, non-interactors of RPA70, such as ATR ATRIP and the mRNA complex, were strongly enriched in the wild-type pull-down. The addition of a separation of function mutant to our screen allowed us to identify the PRP19 E3 ubiquitin ligase as a novel interactor and modifier of RPA, which regulates ATR ATRIP activation and the DNA damage response.